Hello, everyone. Welcome back to the AAS YouTube channel. And this is part of the most awesome good stuff. This is the AAS Journal Author Series, and I am super happy to have Rachel Slank with us today. Hey, Rachel.
Hi, how are you doing today?
I'm doing very awesome on this July 22nd of 2022. Hey, that's a lot of twos in there on that one.
Yes.
Doing well. I'm in Phoenix, and our monsoon season is rolling pretty good. We'll probably get our lightning and thunderstorms and all that kind of stuff later today or tomorrow is when it's rolling in. And how is the weather on Mars, apparently?
I wish I was on Mars. It'd be so much cooler than being in the humid Arkansas heat.
It would be cooler.
Yes. I miss monsoon season. I used to live in El Paso, and I greatly miss those random monsoon storms. But no, just Arkansas humidity.
Well, you still get that Gulf Coast moisture, rolling on up. So
it is moisture. Yep.
It is moisture. It just stays in the air a little bit. It doesn't quite fall down.
A little bit.
So,
yeah.
Very cool. Very cool.
Yeah.
And what's your position there in Arkansas? What are you doing there?
I actually just finished my PhD 2 weeks ago. So I'm in the process of moving to Houston to more humidity. I got to stick with that
you just
theme.
Can't get enough of it.
No. So I'm in that fun process of moving and transitioning to a postdoc down in Houston.
Very cool. Well, congratulations, Dr. Slank.
Thank you.
Appreciate saying that. It's so nice.
It has a nice ring to it. I'm not used to it yet, but I love it.
So let me ask it this way. Dr. Slank, what do you like to do for research?
Well, I love planetary research, but I'm a geologist at heart.
Cool.
So all the geology. Did a lot of moon research with my master's, but wanted to take a whole different route to Mars, and I've now loved both of them. I like to joke that I like the M's.
Yeah, there you go.
But yeah, so I like to stick more to planetary geology as my main focus.
I think Mercury is in your future.
I think so too.
And that
has water. It's fine.
It does. It does. Has ice even. So very cool. And that is going to bring us to this very lovely M article. Yes. One of the M's. And this is a Planetary Science Journal article, and it's open access, so you can get it for free. So go ahead and grab it, everyone. And we have, excuse me, experimental constraints on deliquescence of calcium perchlorate mixed with a Mars regolith analog. And Rachel, Dr. Slank, take us away.
Perfect. Thank you. Yeah, so real quick, I just want to give a shout out to my co-authors, Dr. Edgar Rivera-Valentin, as well as my advisor, Dr. Vincent Chevrier here at Arkansas. So I just kind of want to talk a little bit today about one of my chapters of my PhD and now a paper, where we did experiments in a Mars simulation chamber to understand the water cycle a little bit better on Mars, especially with calcium perchlorate when we mix it in with a regolith. So in this case, it was JSC Mars 1.
Okay.
So just jumping right into the introduction, water is kind of important, right? Water is the key for life. Everyone wants to look for it, and Mars has obviously been a big focus on that because it's close. We know there's some water there. Great focus area. Indeed. But the temperatures don't really allow pure liquid water, right? Kind of is not fun. But what you can do is add salt to it, and the salt will lower the temperature, which will then allow for that water to last a little longer. That's why we put salt on the roads. That's why the ocean doesn't freeze as fast.
Cool.
It still freezes, just not as fast.
Right.
And we know that there's salt on Mars, right? Viking has found some, but the big one was Phoenix when they found some perchlorates. And what's cool about perchlorates is they can get really cold and still stay liquid. In fact, calcium perchlorate is 198 Kelvin before it freezes.
Ooh, wow. Okay. Thanks.
Right? So ideal salt, which is why we're testing it in the chamber.
Okay.
So one of the main ways that salt can lead to this brine solution or liquid water is a process called deliquescence. So you have salt on the surface of Mars, and the salt absorbs water from the atmosphere to the salt. So why we put rice in our salt shakers. Salt loves water.
There you go.
And so that water adsorbs onto the salt, and eventually it starts to dissolve the salt and then leaves a pool of liquid water.
Good.
And then that water will then go and cling on to the next thing, whether that is more salt to dissolve or whether it's the regolith around it. And when that regolith adsorbs to the water, it gets wet and leaves this darker color. And that's a really big indicative sign that deliquescence has occurred due to that regolith change.
Got it. Cool.
So our main focus was kind of just to study this regolith and calcium perchlorate and see how it works, right? We wanted to study stuff in relatively low humidities and see if deliquescence can occur in a reasonable time span. And then if it can, do we see that deliquescence and regolith darkening?
Got it. Yep.
So that was kind of our whole focus area.
I'm with you.
Cool. So all of our work was done at the University of Arkansas in the Mars simulation chamber. You can scroll down to figure 1. This is Ares. He's my best friend and mortal enemy. You know, they always say
tribulations of a PhD thesis. Yes.
They always say experimental work is like 90% getting stuff to work and 10% collecting data. And
yeah,
whew, they were not wrong.
Yeah, there we go.
We're not wrong.
Okay.
So there's a picture of Ares on the left. It's really cute. I dressed him up as a minion one year for Halloween. But Ares is about 2 feet, 3 feet tall and 2 feet in diameter. As you can see, it has a removable lid, so you can actually physically put stuff inside the chamber.
Okay.
As I said, it's a Mars simulation chamber, so I can get it down to Martian temperatures, atmospheres, and pressures. All the fun things.
Yes.
So you can pull it to vacuum, or not vacuum, sorry, 3 millibars of pressure. Although for these experiments, we keep it at 6, since that's the average pressure of Mars.
Okay. Good.
You could input CO2 into the chamber, so that way it creates a Martian atmosphere.
Okay.
And then we can cool it down with our chiller system. In these cases, we kept it close to 0 Celsius, but it can go colder.
Okay.
So I'm going to push you all the way to C. Inside the chamber, we have a bunch of instruments, and I'm not going to talk about all of them, but the main ones I want to focus on is that blue rectangle. I love how I'm pointing, like I can actually use it. The bottom one.
Bottom one, there we go.
Bottom blue.
Bottom
so that
blue.
is our scale.
Mm-hmm.
It takes and records mass every 2 minutes, and our sample actually physically sits on top of that.
Okay.
We have 4 thermocouples to measure temperature, 1 physically in the sample, which is that fun red rectangle, 1 right above our humidity buffer, 1 in the upper atmosphere and 1 in the lower atmosphere.
Cool.
As I mentioned, we have a humidity buffer because even though we're putting in CO2 into the chamber, we want to also control some humidity. So in these cases, in these experiments, we used a lithium chloride saturated solution. So I take distilled water and I throw a crap ton of lithium chloride salt in there until it becomes saturated.
That's a technical.
And that helps control the humidity to about 11%.
Okay. Okay.
We also have 3 hygrometers inside the chamber that measure relative humidity. Again, 1 above the humidity buffer, 1 above the sample, and then 1 in the upper atmosphere.
Yep.
So trying to collect a bunch of data points. We also took pictures before and after the experiments to see if we see that regolith darkening, which we'll kind of see in a little bit with this paper.
How long does one experiment take? Like, how long do you let this sit in here? Is this measured in minutes, or is this hours or days, or?
Yes. So it really depends. Most of our experiments were about 6 hours, but I did run 1 for 24 and 1 for 48.
Okay.
But it takes a little bit to prep it because you obviously want the chamber cold and everything set up. You also want to pull the chamber to vacuum and then infill it with CO2. That way there's very little water in the chamber to begin with because you're trying to see if water goes into the sample, right? So you don't want it contaminated to begin with.
Right.
We also make sure that there's no water in our physical samples. So we bake our JSC Mars 1 and then we put it in a desiccator for 24 hours and pulling it to vacuum. And then we freeze it to make it cold because Mars is cold. And then with our salt, we also put it in a desiccator for 24 hours and then the freezer. That way there's as little bit of water as possible.
Good. Okay.
And then right as the experiment starts, we open the lid. We quickly mix the JSC Mars 1 and salt and put it in the chamber before closing the lid and pulling it back to vacuum. Cool. And I say quickly because obviously as soon as you take it out of the desiccator, you're exposed to Arkansas humidity, water.
There you go.
So we try to make it as fast as possible and eliminate as much as possible.
Cool. Good.
But, you know, science.
Science.
So going down to figure 2.
Okay. Ooh, in action here.
Beautiful. Yeah, so you saw the fun diagram, but it's always better just to see it in real life. So you kind of see the scale with our sample sitting on top of it. That reddish brown color is the JSC Mars 1. It's a Martian simulant that we used. And then the white is calcium perchlorate.
Yep.
For these experiments, we did anywhere between 0 and 5.5 weight percent. Trying to make it as Mars relevant as possible. You see the lithium chloride buffer and right over there. And you see that I physically have taped thermocouples into both of those. And then there's 2 more hanging out in the chamber.
2, 1, 2, yes.
Yep. And then there's 1 in the upper and lower atmosphere that aren't in this picture. And then you also have the hygrometers that when the lid lowers, it'll then be in position to be right above the buffer and the sample and then upper atmosphere.
Okay.
They don't look it in this picture, but as soon as the lid closes, they kind of go into place.
Yes. Yes.
Yes.
The cord there coming off. Okay, cool.
Right. Yeah. So that's inside the chamber right before it starts. And then if you scroll down to table 1, I can kind of walk through our experiments. We don't have to focus on all the fun details on it, but we did 11 experiments, 6 of which were controls, and then 5 were actual mixing of the JSC Mars 1 with calcium perchlorate.
Cool.
So there's 2 sets of controls. Each set had just an empty petri dish, one with just the JSC Mars 1, and then one with the calcium perchlorate.
Yes. Okay.
One set had the lithium chloride buffer in there, so we see it at that higher humidity, and one that was kind of just chilling without a buffer. So some have a higher humidity than others.
Yeah.
Okay. Once we did those fun ones, we then actually got to have fun and mix stuff together.
There you go.
So all of our samples had 2 centimeters of JSC Mars 1. And then, as I said, we ranged between 1 and 5.5 weight percent of calcium perchlorate with these. We lightly mixed it on the surface. We originally had mixed it completely and then realized we couldn't actually see what was happening below. So we were just like, well, that's a waste of salt. Let's just put it on the surface.
There you go.
As I mentioned, right, we mainly aimed for 6 hours. And then experiments 10 and 11 are longer because we're like, well, let's see what actually happens. So one was for 24 and then one was for 50 hours.
Okay.
Those are the bases.
Yeah. Okay. Very good. Yeah, answers my question.
Yes. So rolling down to figure 3.
Protocol. Ooh, stars.
Right? It's space themed. You got to have stars. And of course, they have to be neon. So as I said, one of the first things we did was a bunch of controls to actually see if this works, if these parameters work, all that fun jazz. So figure A and B is the calcium perchlorate control with the humidity buffer, and C and D is without the buffer.
Okay.
As you can see, the before and after of A and B is much different than the before and after of without. Right? Right? The stars are just there to show you that these are the same salt crystals and that I just didn't pick random stuff.
Right. Okay.
Right. But so you see in figure A, the salts are really white.
Yes.
They're really opaque and you can't really see like the bottom of the petri dish. And obviously in B, everything's more translucent. It's shiny. You see the bottom of the petri dish, it's slushy. Right.
So darker. Okay.
Yeah, well, that actually isn't darker. It's the bottom of the petri dish with my hand with the glove.
Right. Understood.
Yep. So we obviously see that the salt has deliquesced at least to some extent there. And that was with the humidity buffer. But in C and D, when the humidity was much lower, we really didn't see a change. It was really opaque in both of them. The salts are about the same size. Nothing happened.
Yeah.
Right, which is good, right? It shows that when I add the humidity and make it a little higher, right, that there's enough water for deliquescence to occur in the chamber.
Cool.
Right, so then we're just like, yes, okay, we can actually do real experiments now. So going down to figure 4, right, one of the main goals was just to see if deliquescence could happen in these conditions. Right, so we plotted all of our experiments on a phase diagram. For water with temperature on the y-axis in Celsius and relative humidity on the x, and then everything's color-coded. And there's that bright blue line, and that's called the deliquescence relative humidity line. And when you cross over that line into the gray, it is showing conditions where liquid can occur.
Cool.
And as you can see, right, you're circling 5 experiments where deliquescence should occur,
Mm-hmm.
right?
Yep.
And there's one right on the cusp of that deliquescence relative humidity line that we're going to say can't produce conditions, and then one off in the middle of the white that is like, nope, that can't happen. And
Okay.
that nope one is actually that calcium perchlorate control that we saw up in figure 3, right? So
Yep.
nothing happened there. Hey, look, it also says that it was not in a condition.
Got it.
Right. So the first thing we did is we kind of looked at that experiment that was on the blue DRH line, right? It should not see deliquescence occur. And so we wanted to kind of examine that further. And so if you look at figure 5.
Let me ask 2 questions on this one.
Yeah,
What is the red line?
It is the efflorescence line. So that is the opposite as you're drying out the salt. So deliquescence wets the salt.
Yeah.
And the efflorescence dries the salt.
With you.
Yes.
And the color coding is it's just a experiment number.
Yes. Yep. I just picked colors to make sure they were visible and different.
Yep. I just did want to make sure it was some physical quantity. Okay. Very cool.
Nope. Just wanted to make sure that everyone was like, yes, experiment 11 is bright yellow. Cool. But 5 is purple.
Very good. I'm with you. Good.
Yeah.
Okay. And figure 5, you were moving on to. Ooh, here we go. Experimental results.
Right. So this is the one where we did not see where it should not have allowed deliquescence. And you'll see in green is mass and blue is the change in the water vapor pressure. And both of these are over time. In this experiment, it was about 4 degrees Celsius, and we had a relative humidity of about 10% in the chamber, and it was 1 weight percent of calcium perchlorate. So the main thing I want to show with this water vapor pressure diagram is that it's positive, right? And so what's happening is there is more water in the atmosphere of the chamber than there is in the sample. And so in order for everything to be in equilibrium, the water vapor needs to go from the atmosphere into the sample. And as that happens, you would gain mass,
yes,
right? Because water is physically going into the sample. Cool. It's what we see in that green line.
Yes.
Okay. So I'm actually going to jump you down to figure 11.
Figure 11. Let's go to figure 11.
Right. Scroll all the way down.
Keep that figure in mind, and we will go to figure 11. There we go.
Yeah, I wanted to jump around a bit.
All good.
Righto. So these 4 are 4 of the experiments that we should have seen deliquescence occur when we were looking at that phase diagram.
Okay.
That 5th point was actually that other calcium perchlorate control that we saw, that obvious change. So these are the other 4.
Okay.
And so with these 4, right, they're all between 4 and about 5 degrees Celsius. You'll notice that the humidities are much higher on these, right? Around 19 to 22 relative humidity. So, and then our weight percent of calcium perchlorate is still 1 to 5.5.
Okay, I'm gonna blow out to get a global.
Yes.
Okay.
Right. So you see that. So same setup, but
Yeah.
one thing that is different, and you can zoom into any one of these.
Let's take A.
Perfect. Is now you'll notice that the change in water vapor pressure is negative. And it's actually negative for all 4 of these, anywhere between -50 and -20 pascals.
Okay.
Which means the exact opposite's happening in the chamber. There is more water in the sample than there is in the atmosphere.
Out gas.
Right? So in theory, if you wanted it in equilibrium, we should be seeing a mass decrease because it should be leaving the sample to the atmosphere.
Yes.
But in all 4 of these, we see a mass gain at least somewhere in the experiment. So A is the only weird one that kind of increases and then decreases.
Right.
Although I want to point out it's very little. It's right. You're seeing 267.7 to 267.4. So 0.3 grams. So not a ton. But if you go over to B, C, or D, you'll notice that throughout most of the experiment, it is increasing in mass. It's kind of weird, right? Because
larger amounts too.
Yes.
Yeah.
A just wanted to be a little difficult and unique. It's fine.
It's experiment. It is. Ooh, and here we have a sawtooth. Okay.
Yep. So even though the chamber should be going towards equilibrium and losing mass, that is not what is happening. Right. Which means something is controlling the water vapor at the sample. We believe that process to be deliquescence.
Got it. Mhm.
Like, cool, okay, these should see deliquescence. We're seeing some evidence that this is occurring. This is a good sign. So then the next step was to go and see if we saw that regolith darkening, right, which is indicative of that brine completely dissolving and then clinging to the dirt. Sorry, the regolith. The soil people do not like it being called dirt. It is
regolith.
It's regolith. I took a soil physics class, and that was like one of my hardest things to transfer. That dirt, soil, and regolith are very different,
Very
and
different
certain people have an issue.
And
So, as I said, that was our next step, was looking for that regolith darkening.
okay.
But we did not see it in any experiments, which is a little disappointing. And so we wanted to go and look for evidence of deliquescence, even though we weren't seeing that. So yes, I see you jump to figure 7. This is actually after the experiment has occurred.
Okay.
So I'm going to quickly jump you back up to 6 right before we talk about that.
All good.
Yep, no, so we didn't see any regolith darkening, but we still wanted to kind of examine everything and see if we could find other signs of deliquescence occurring. So when we, as I said, we have all these instruments inside our chamber, and I'm just going to briefly touch on this, but you'll notice that at figure part A, I guess, you'll see that mass increase.
Yes.
In B, it's relative humidity over time. That orange color is our sample.
Okay.
The blue is upper atmosphere, the green is the buffer, and then the red that is only in the temperature is that lower atmosphere.
Yes. Okay.
Right. So the main things I want to point out is that the orange, or the sample, has a much higher humidity than the rest of the chamber. And that actually is the case in all of these experiments where we should have seen deliquescence occur.
Okay. Okay.
And that in the temperature, by the end of the experiment, our upper atmosphere and our sample are the same temperature roughly.
Yes.
Followed by then the buffer, which is a little cooler, and then the lower atmosphere. And so temperature does affect relative humidity, right? And so that also explains a little bit of why our relative humidities are varying, but not completely, right? Because our atmosphere and our sample are the same. I love how I'm pointing. You still can't see my pointing.
I'll point.
Right. So they're still the same, but obviously the relative humidities are not. And so part of that is that deliquescence and the salt.
Yes.
Right. So anyways, in all of ours, we saw that mass increase, and we saw that our sample had the highest relative humidity in all of our experiments.
Okay. Interesting.
Yes. Yes. So jumping down to 7, which you had pointed out. Right, this was during one of our experiments. It was experiment 9. So it had about 5.5% of calcium perchlorate. And as I had mentioned, we did not see any regolith darkening occur in any of our experiments.
Okay.
However, when we took it out of our chamber and exposed it to the loveliness that is Arkansas humidity, we really quickly saw deliquescence occur. So in figure A, there's nothing. Sad day. Right, I pour this sample into a different Petri dish because that one's kind of attached to the chamber. Right, and I just left it on a table, and within 5 minutes, you start to see that regolith darkening. You see those dark spots kind of in the middle.
Yeah.
And then by 30 minutes, you see a nice amount of the salts deliquescing.
Cool.
An interesting fun point that someone should research later on is that there does not seem to be a rhyme or reason to why some of the salts deliquesced and some didn't. Right, so you'll notice and see that there's some big crystals that deliquesced and some little ones that deliquesced, but you also see big and small salts that did not. So that was really interesting, and we noticed that throughout our experiments, that it didn't really matter that the grain size of the salt.
Well, that's interesting.
Right, someone should focus on that. Yes, I also want to point out, since I'm sure it will be asked at some point, we did not grain size control our calcium perchlorate, right? It's not controlled on Mars. And so we just kind of kept it within a small range, right? We didn't just like throw half a Petri dish of salt in there. We did break it up a bit, but we didn't want to be like only this size, because then you wouldn't have seen these fun, weird effects.
On particular grain sizes, right?
Right. So just wanted to point that out.
Okay, good. Thank you.
Yeah, of course. So scrolling down, you can briefly show figure 8. It's really just showing the same thing I already said. We saw a mass increase. Our
Right.
humidity is higher above the sample, and
Yes.
our sample and upper atmosphere by the end are the same. Same fun
Okay.
conclusion,
Yes. Same.
right?
Uh-huh.
And then if you scroll to figures 9 and 10,
Do
it
not.
is just showing more examples that we did not see that regolith darkening in the chamber,
Right.
right? Although with figure 9, we did see some weird droplets on the side of the petri dish and a little bit of regolith darkening at the very, very bottom.
Yes. Let me
Which
pull
we
it up
found,
a bit.
yeah, we found it really interesting, right? None of the salt on the top Deliquesced,
Ew. Okay.
right? But at the very bottom, when we dumped it out, there was a little bit of wet regolith on the bottom.
Mm-hmm.
So we're still trying to figure that out. But because
Yeah.
none of the salt Deliquesced on the surface, which is what we were looking at, we also wanted to just state that we're claiming this as it did not show that regolith darkening.
Right.
But again, you
Yeah.
dump it into the petri dish, and within a few minutes, we're seeing that regolith darkening.
Okay.
And you'll notice this one was about the same time in E as it was for the previous one, and there's much less deliquescence occurring.
Yes.
What is really interesting, and I don't have a picture of it here because it was kind of boring, but the experiment that was on that DRH blue line, the one that should not deliquesce,
Yes.
did not deliquesce after I took it out of the chamber for at least the hour that I was staring at a
Wow.
petri dish regolith, right?
Okay.
Also, the 1% weight calcium perchlorate that should have deliquesced also didn't show any regolith darkening after I took it out of the chamber for a bit.
We have many mysteries here. Okay.
So we actually believe that the reason that is happening is because there was not enough water. There was not enough water inside the chamber for that regolith darkening to occur.
Okay.
Right. And so there's very limited water. That water is competing to go into the salt or to be absorbed into the regolith itself.
Right.
And obviously a little bit of deliquescence is occurring, but not a lot. A great example of that is, no, did I maybe not include it in this one? In my PhD defense, I actually had a really cool picture of figure 10. So if you want to blow up into A, it's not as much fun because I can't show you the before on this one, but some of these salts, especially at the very bottom rim, actually are a little bit smaller than when we put it into the chamber. So the salts are a little bit more transparent here and a little shinier.
Yeah.
So we know that the salts are slightly deliquescing, but not to the full extent that we would have liked to see.
Right.
And so we think that the salts are starting to deliquesce and that some of it is getting absorbed,
Okay.
but there just was not enough water vapor inside the chamber.
Okay.
And in fact, we did some fun math where we tried to see and kind of prove that's the case, right?
Okay.
And so in experiment 10, which is the figure 9, right? If all of that salt had deliquesced,
Okay.
right?
Okay.
We should have seen 12 grams of a mass increase because all of that salt should have turned to water.
Yes.
But we actually only saw about 1 gram increase of mass.
That was the delta on those y-axis. Yep. Mm-hmm.
Yep. And so if that 1% only was due to water going straight into the salt, which is not realistic, only about 8% of that salt should have deliquesced.
Okay.
Right. So we
Interesting.
just are seeing that there is just not enough water inside the chamber, which
Yes.
I guess makes sense, right? Higher humidity, more water, so more salt can deliquesce.
Correct.
We
Yeah.
also, right, I pointed out a few times that mass increase,
Mm-hmm.
right? Because water is going into the sample, whether that is the salt or the regolith itself.
Uh-huh.
And we noticed that in the experiment, the control that was with the humidity buffer, but with the just pure regolith,
Okay.
we saw a mass increase in that as well.
Okay.
Which is cool. But we saw it increase about 22 milligrams per hour.
Okay.
Right? So a little bit, but not a lot. But when you mixed salt into that mixture, it doubled in mass increase.
Mm-hmm.
And then if you had it with the experiments with 5 weight % or more, it actually increased by about 52 milligrams per hour.
Okay.
So
Sure.
quite a difference than that 22.
Okay.
So it's interesting. A lot of people were worried that deliquescence and adsorption would actually compete with each other, but it is actually showing in these experiments that kind of work hand in hand. And so more water is being adsorbed into the sample when salt is present.
Yes.
Which is really cool.
Very nice. Very nice.
Yes. So that is roughly it, right? So we can jump down to conclusions. There's no pretty pictures there, right? But our experiments basically showed that deliquescence can occur in these conditions, right? We saw that with that negative water vapor change, right? Where we should have seen a mass decrease, but we saw that mass increase, right? We actually saw the increase happens quite a bit more when the salt is added. And then we saw physical changes to the salts themselves. And then we saw the salts deliquesce very fast once it's out of the chamber. But we did not see that regolith darkening that was indicative of deliquescence, again, because there just was not enough water vapor inside the chamber. But, you know, it's experimental work, so more experiments are always needed with this. But it is still really important, right? Because we want to understand that full range of deliquescence on Mars, right? And in Mars-like conditions. And on Mars, you range anywhere from 0 to 100% humidity, right? So it's nice to at least see it here in the cases of 20% humidity and then be able to compare it later to higher humidities to see if we can get that.
Right. Okay.
Yeah.
Very
that
nice.
is it.
Very nice.
Yes.
Very cool. Rachel, thank you so much for walking us through this very awesome PSJ article.
Of course, thank you for having me.
Well, yeah. You mentioned it a couple times, or you alluded to it a couple of times. And so let me ask it is, you know, where do we go over the next, say, 2 to 5 years? Clearly, I think there's more experiments to be done.
All the experiments.
You know, but are there other things? Are there theoretical calculations that could be done on deliquescence? Are there additional observations maybe coming from one of the Mars missions that can shed some light on this? And just sort of, you know, where do we head over the next, let's say, 2 to 5 years on this topic?
Yeah, so start with the experimental. Obviously, everyone wants more experiments, and there's so many like fun things you could tweak. Right.
I had ideas as you were talking about it. I was going, well, wait.
Right. So one of the main ones is obviously to test this with higher humidities in general, right? We want to see that regolith darkening occur.
Yeah.
So that way we can kind of fully understand that process. We also should test this at lower temperatures.
Okay.
Right? Because Mars, even though 0 is really fun, Mars is not usually 0 degrees Celsius. It's much colder.
Yes.
So that's another fun set to test. So there is a fun span. You could test different weight percent. You can test different salts. There's a lot of salts on Mars, not just calcium perchlorate. Might be fun at some point to throw a bunch of different salts into the chamber and kind of see. There's also different regoliths you can test. Just like how I said, people have a preference of dirt versus soil and regolith. There are apparently strong opinions on what regolith simulant you use. And I've learned no matter which one you use, people are going to have strong opinions on it. Somebody will be upset, yes. Yep. So there's a lot of fun parameters and variables you can tweak there. On the modeling side, right, a lot of people want to try to model it, not just with the salts, but how that reacts on Mars. So Riviera et al. In 2020 had a paper where they showed how long salts could last throughout a year on Mars. And so with calcium perchlorate, there were spots where it could be up to 2% of a Martian year that liquid water could remain stable.
Okay.
It's pretty cool. Not a lot, but it's still cool. And so there's a lot of modeling we can still do, again, tweaking the salts and the parameters to kind of see, is this viable? On the exploring Mars side, it's kind of tricky, right? As I mentioned, salt allows water. Water is important. But as soon as you have water, you have to do planetary protection, because what if we bring microbes or bugs or something when we're exploring? People, right? And so sadly, right, we're not just going to go and land where we think there's water, because then we might contaminate it. But rovers are still exploring the surface and seeing salts. So it'd be kind of nice to see what those rovers find with salt mixtures, salt compositions, percentages, right? We might find a giant clump of a certain salt that's more than 5 weight %, and that'd be an ideal spot to look in the future. But it's kind of still that catch, right? Where as soon as you find it, you're supposed to go the other way because contamination.
We're not
right.
We're not masked up yet. Let's put it that way.
Exactly. But yeah, no, there's a lot and so many different variables that you can test for the next few years and beyond.
Cool. Well, I look forward over the next couple of years to seeing some of these mysteries, and there were several, start to get resolved as we do more experiments and all that kind of stuff. So
me too.
I look forward to your next article on the topic. So very cool. All righty, Rachel, thank you so much for walking us through your very lovely article.
Of course, thanks for having me.
Sure, my pleasure. And that will do, everyone. And I hope this made your astronomy day just a little bit better, and we will see you on the next one. Bye-bye.
Bye.